Yes. Then I went back to the lab. I got it back to the lab because I got lost. Which would have been way better when I originally.
But this is a lot older.
Well, that's why I brought coal.
Yeah.
Then welcome everybody again. I'm glad that almost everybody is here again. This time we have as our speaker Terry Sejnowski, who just flew in this morning from San Diego. So if he starts mumbling a bit, then it's because of jet lag, but I'm quite sure he will not. One of the nice things of Terry and Sydney together is they both know very much the same people. There's a lot of age difference between them, though they look at those people from a different point of view. Terry will talk about neuroscience and the influence of the molecular biology on neuroscience. I think that's the topic. But it will be in the interaction between Sydney and Terry in the end that will make the whole thing going again. And so I will not make any further introduction because the best thing to do for you is to talk. And so I hope you enjoy yourselves like you did in the last two times, and we'll be having questions and answers after your talk.
Well, thank you very much for inviting me, and also thank you, Sydney, for those two marvelous lectures, which I enjoyed coming over on the airplane. So they sent me the videos, so I was able to go through and appreciate all the amazing perspective that Sydney has on the history of that era, present at the creation. Now, Sydney and I go way back, but one of the things that you have to understand about Sydney is that he's probably was the most peripatetic person I knew. So not only, of course, have this original group at the LMB in Cambridge, but he also, it turns out, and this didn't come up in the lecture, he was the founding president of the Okinawa Institute of Science and Technology in Okinawa, Japan. And he organized, it got it off the ground, and it's doing extremely well, and it's really quite remarkable because he set it off on a course that was very different from the traditional Japanese university, which is very top-down. His perspective was, it's really the science, as you heard in his lecture, comes from the bottom up. It comes from the youngsters, the people who are looking at a problem freshly for the first time. And so that was an important part of the way that that university got off the ground. And by the way, founders like Sydney really have an enormous impact, not just on institutions, but on whole fields of science. For example, there's a field which, as you know, he started C. elegans research, but that group is the most cooperative group I've ever come across in terms of sharing insights, reagents. They really did all spring from your group, and now they are in four corners of the world. In fact, I have a colleague right next to me who's, I think, third or fourth generation, who's working on C. elegans. But the other thing, if that wasn't enough, right? So he was also a senior fellow at Janelia, which he mentioned. He was also had a lab here in Singapore, and you came here regularly, and you mentioned this, right? So, my goodness, where else? It turned out that every time that he would go between Cambridge and somewhere in the Far East, he would stop in La Jolla, where he had a condo overlooking the ocean. And he had many friends. In fact, he was even a distinguished faculty at the Salk Institute and on the faculty of the Scripps Research Institute. So, I mean, he really managed to like Johnny Appleseed, sow ideas between all these different institutions, and it's really quite amazing. So the role has reversed, and over the last year, I've visited Singapore 3 times. So maybe I'm the one that's become peripatetic. So let me tell you this one story. We're having dinner in La Jolla at your favorite restaurant, that Italian restaurant. And so I thought I would tell Sydney the story I had heard back when I was a graduate student. So I mean, that was decades ago. And so I told him this it's kind of a shaggy dog story about Francis Crick. So the story starts out that Francis Crick dies, and he knocks on the pearly gates, and St. Peter greets him and says, "Oh, Francis, what a surprise. We didn't expect you here." Of course, Francis was a devout atheist. And Francis says, "Well, all my life I've wanted to know the answer to this one question." And St. Peter says, "Well, of course, Francis, you absolutely deserve to know the answer to your question." So Francis walks across this field. This field is filled with gears and wheels that are rusting and, failed experiments. And he knocks on the shack. And there is this old man in a leather apron, clearly in the middle of an experiment. He has grease on his hands. And he says, "Oh, Francis, how wonderful. What can I do for you?" And Francis says, "Well, all my life I've wanted to know the answer to this one question. Why do flies have imaginal discs?" And God looks up in surprise. "Why, Francis, no one has ever asked me that question before. I've been putting him into flies for 200 million years, and I haven't had a single complaint." Okay, so I'm telling Sydney this story. His jaw drops. And I said, "Oh my God, this is one of his best friends. I mean, my God, this is really awkward." And Sydney says, "Terry, I'll tell you the moment that story occurred to me." And now let Sydney tell his version and where the story came from.
Well, you see, Francis and I shared an office for many years in Cambridge. Above Francis's head, there was a big sign, which he got actually from a friend of ours, it said, "Reading rots the mind." That's the story. That was actually a piece given, and all the time Francis just sat there, surrounded. Indeed, when I first went to see him in La Jolla, there was the same sign, but every surface was covered with copies of papers and everything. So Francis decided, as you know, Drosophila is developed from a set of, they're called imaginal discs, and one develops into legs and one develops into antenna. And Francis got very concerned about this. So, very frustrated, he banged the table. He said, "God knows how these imaginal discs work." And at the moment, I had the story. I, in fact, went to Oxford to give a lecture, and I put the story in the lecture. So Francis eventually goes to heaven. He's met by St. Peter there, who says, "Welcome, Dr. Crick." He said, "Please sit down, have a drink, relax." He says, no. Where's this fellow God? I've got to ask him a question." So they said, okay. So they take him from the front part of heaven, right through the middle part of heaven, right to the back part of heaven, where there is this shed with a corrugated iron roof. And outside there just is all bits and pieces of everything lying around. And they go in there, it's quite dark inside, and there is this little guy in a leather apron, as Terry tells the story, working at the bench there. And he says, I'd like to introduce Dr. Crick to you. He said, God, this is Dr. Crick. And Francis said, oh, I'm delighted to see you. I have to ask you this question. How do these imaginal discs work? And God looks at him, thinks for a while, and he says, well, we kind of took this stuff and we added it to that stuff, and that other stuff got incorporated in. He pauses and he says, actually, we don't know how they work. But let me tell you one thing. We've been building flies here for several 100 million years, and we've had no complaints. So that's the story of the great mechanic in the sky, the garage mechanic.
And what was the title of the story?
It's been published, and I can't remember for the moment.
Oh, okay. So this is Francisco Crick.
Oh yes, it's called Francisco Crick in Paradiso, because it had to be Italian, you see. Francisco Crick in Paradiso.
So what really floored me was that, I've been retailing this story for decades, and to actually find the origin. Now, what's interesting, you'll notice if you compare the two stories, the details have all mutated.
Yes.
Right? But the core of the story is the same, the punchline is the same, and this is something, when a story has gone through several brains, this is what happens to it.
Well, the punchline of the story is you can know a lot to build things. You don't have to know how they work. In fact, he didn't know how they worked.
Now, so I have had did an experiment because this is something that I've told to many people, friends, and I've noticed that there's a dichotomy between my physics friends, because I started out as a physicist, and my biology friends. All biologists immediately understand, and they just get it immediately. And the physicists just stare. They have no understanding of what the fundamental biological principle is, and what's an imaginal disk. What's the point of this story? So it's really the two parts of my background really had it really you have to integrate the what where you came from and where you're going. So Sydney wanted me to tell you a little bit about myself, and it actually reminds me of the time that I gave a distinguished lecture at the University of Texas at Austin. And my host gets up in front of a big audience and says, Terry Sejnowski needs no introduction. So let me tell you a little bit about myself. He eventually got back to me, but here's where I got started. So I was a graduate student at Princeton studying relativity with John Wheeler. And this is something Sydney brought up in the last meeting. And it was a really exciting time because John Wheeler had just coined the word black hole, and they hadn't been discovered yet, but very exciting that you can actually look for them. And that really was what his strength was, that he took some mathematics and then asked the question, well, if it exists somewhere in the universe, where should you look to find it? And the other thing that was very exciting back then, in fact, I was working on it for a master's thesis, was gravitational waves, which, we're back at this echo. Gravitational waves, which at the time there was a group at the University of Maryland who thought that they had discovered them, turned out to be a computer bug, a program that was supposed to analyze the signals coming from these cylinders. And so what I did for my master's thesis was to compute expected signals from all known sources, supernovas, colliding black holes. Those are the two big ones, and then a lot of other possible things. So you need to have a big collision. A huge amount of energy has to be released because these gravitational waves are very, very weak. And what I computed was that we were at least 3 orders of magnitude away from a known source. You might get lucky, maybe there's some other source you don't know about. And I figured that it would take at least a decade for an order of magnitude. So that meant that I would have to wait 30 years for the first sighting. Now, what's remarkable is that, almost on cue, last year, gravitational waves were discovered. And in fact, the paper, that founding paper, which was published in Phys Rev Letters, had 1,000 authors on it. Just as Sydney mentioned now, big physics, because it involved massive amounts of, it was the biggest NSF project ever, hundreds of millions of dollars, right? It was just huge. And of course, it's very important, but what I realized back then was that I couldn't wait 30 years. I'd be an old man. In fact, I am. But I was taking a couple courses in biology and in psychology. I took a neuroethology course from Mark Konishi, who studies barn owls and sound localization and songbird song learning in songbirds. And it was fascinating because this is just as mysterious as the cosmos, but it was something that you could actually study with your own hands. You didn't need to have a supernova go off. You didn't need to have a $200 million project. And it was really exciting. And the other course I took was from Charlie Gross, who was at the psychology department. And this was all about vision, the visual system. He studied the monkey visual system. And so I was reading Hubel and Wiesel's papers, just the same ones that Francis was reading. And that was also very exciting. And that's when I decided that, I would really like to become a neuroscientist. Now, how do you do that? I did finish up my PhD in physics working with John Hopfield, but I was very fortunate on 2 counts. One, I took a summer course at Woods Hole, the Marine Biological Laboratory, and this came up also in some of your discussions, where I was trained by some of the best neurobiologists in the country, and as a consequence of that, I was offered a postdoctoral fellowship with Stephen Kuffler, who was, at the time, his department of neurobiology at Harvard Medical School was the leading place for neurobiology, and that's how I got trained. It was very, very exciting. And the problem, however, was that the further I got into the brain, I was recording from ganglion cells and trying to understand the signals, molecular mechanisms, and so forth. And the further I got in, the further I realized that there's an infinite amount of detail here. And, how do you choose? And this is the problem that Sydney has been spectacularly successful as. What is the right question to ask? Right? You want a question which, if you answered it, you could really make a big advance, but it's not so complicated that you won't be able to. And I just wanted to ask Sydney, what's your secret?
Talk to the right people. Yeah, I think it was, everybody really understood that genetics would be at the core of biology. But genetics had got to a point at that time where it was in the hands of the people who were doing, theoretical genetics, Fisher, and a whole group of people just not studying the gene. And I think that we wanted to get to the bottom of that, and so I couldn't do much on my own in South Africa. And so what I decided is I would just teach myself how to do things. So I taught myself organic chemistry. I used to synthesize my own dyes, and I did a whole lot, and that's a tremendous background that you can just turn your hand to everything. What I was not good at was mathematics. I couldn't follow it if I read. I love geometry. I'm quite a good geometer, but all this algebra stuff was beyond me. Fortunately, I had a good friend who was a contemporary of mine called Seymour Papert. You may have heard of his name because he's the one that landed up at MIT working with the group there. And it was Seymour, as I've said, I managed to get him a job in the physiology department. But I said I'd only do it if he taught me mathematics. I would teach him neurophysiology. I was quite good at that. I knew a lot about transmitters and the brain itself. So now the question is, I'm still a second-rate mathematician, but Seymour is a good teacher. I'm just a poor student. But he got interested in the brain at that point. And as you know, you will hear as this develops, he became involved with a group at MIT to which there were further connections. Dave Hubel, whom you've heard about, was a very great friend of ours in Cambridge. I tried to get him to come to Cambridge, as I did Max Cowan. These were all good people that you know about. But unfortunately, Britain couldn't pay them enough money. We were all very poorly paid as academics, and we paid 70% of our salaries in income tax. So that's of course, if you have people like the Americans running the world afterwards, and a rather impoverished country, heavily damaged in the war. So, but in fact, it was a great place to do science because there were no walls, and the thing you could go and see anybody, and so we learned how to go and meet people, ask the right questions, and it's Cambridge that had all of these scientific discussion groups. They were societies that we all joined. There were several physics groups, and so we often created these groups. Francis did the same thing, the Helmholtz Club and all of those things. They were very interesting things because there weren't many official seminars, but you had people, you gave them a good dinner, and they spoke after dinner. And that's how I learned a lot of things in Cambridge, by going to the meetings. And Francis did that when he went to the Salk. He helped to form a number of these things. And I think that is still a good habit to do.
You mentioned the Helmholtz Club. So when I moved to the Salk Institute, I became the secretary of the Helmholtz Club.
Yeah.
And it's actually was probably the most formative part of my education was those discussions. And it was a small group. Maybe we typically had 15, 20 people in a small room. We would invite an outside speaker. We'd have to come and give a talk, and we'd give them an hour and a half. And then there'd be a break, and then we'd have a local speaker on a single topic. And it was primarily vision, but the brain in general. And what was wonderful was that these were all super experts. Francis was very interested in vision, but there were experts on psychophysics, Ramachandran, Stuart Ansis, and then there were people interested in neurophysiology, John Ullman from Caltech, these were people from universities that were within an hour drive of UC Irvine, and so they would come there for the afternoon. Then we'd have dinner afterwards, and the discussion would continue. And it was like having a master class because everybody there was at a very high level. And it wasn't just a talk. And in fact, I warned people that they should have their summary slide first because they're never going to get to the last slide. Because everybody would jump in, and it would just be this wonderful discussion. And it probably was the sort of, he imported that from the experiences you had.
He imported that from Cambridge. There was a very famous thing. Many of them had been founded. There was the Del Squared V Club. That was a physics club. And there were several of these. And you could easily come along as a guest of a member and easily become a member. They weren't exclusive. The only condition is you had to be really interested in the subject. So one could hear about everything by going to these. It was one night a month or something like that. So that informal thing, not by institutes or by locations or departments, always cut across everything, because you would know people. So the departments had their seminars, and whether we went there or not was another question. But I know that Horace Barlow and I organized one year. We organized a meeting, a group. It was called Problems of Biology. It was a club, and we said people would come and we'd have speakers there. It was just the very early days of molecular biology. And Horace was a great physiologist, and he was a grandson of Charles Darwin, the famous one, as well as because his mother was Charles Darwin's granddaughter. It was a great-grandson. And they lived near us in the same road. And so we had, and we organized this. And we shared a room in one of the buildings. It's the top of a staircase. And so you had your outer room. And so Francis called me and he said, "How many chairs should I get?" I had no idea. He said, and I suggested 12. We were astounded. 65 people packed the room. They were sitting on the stairs outside. There was such a hunger to hear about new science. And a lot of the people were recruited as graduate students through this Problems Club. And I think this informal education, which is not structured, is part of the real joys of doing advanced science. But now, of course, people used to take the register seminars. You had to attend to get what was called your due performance certificate. Anyway, we had that for some subjects, whereupon what we would do is only a few of us went to these, and we would sit at a place in a bench, fill out the certificate. Then when the lecturer turned his back, you would move your place to another one and fill out another certificate for a friend. I used to register maybe 10, 12 people for these meetings, but however, informal discussions were the best. And whenever I had a lab, there was something called the SB Educational Society. We met every Thursday afternoon, but nobody was given any time. They were told at the lunch, they would speak after the lunch. And all the Americans cried and said, "You gave me no time to prepare my seminar." We said, "Yes, we want to see whether you can do it. You, the expert on this, you don't need any preparation." So the informal, being able to talk about your work on the dot, is very important because it helps to clarify the way you think about it. Sorry, it's a big deviation.
No, that's wonderful. I mean, in fact, I've learned a lot about what it was like to be part of the community in Cambridge at the time and all of the opportunities that the students had. By the way, your discussion about the way that you and Francis really complemented each other in terms of you coming up with ideas and Francis swatting them down, or at least exploring them with you, reminded me of something else that when Francis moved to the Salk Institute back in 1975, I think, or 77, for a couple of reasons. One that you mentioned, which was the fact that he would have been, what's the word in England when you get fired?
Well, he came previous to that on a sabbatical.
Oh, that's right. No, he actually was.
And then decided to stay there.
Right. But I think that there were a couple of reasons. One, he wanted to continue to work, and the second one was that he wanted to switch to neuroscience, which is very difficult to do in your home institution because you're surrounded by the same people that you always talk to, and you really want to talk to a different group, and that was what he did. He just jumped into it in a way that I was just astonished at the energy, and you pointed out he had piles of papers. He would read very detailed neuroanatomical papers having to do with the fine detail of how the visual system is organized in cats, and then the physiology from the recordings. And he did something which was very unusual. He basically, and I think that you were one of the people who helped him in this, he would bring in experts in all these different areas and for a week, and then he would just have this dialogue with them and basically pick their brains and find out all these questions that he had, and there's no better way to do that than have an expert there who can answer your questions, because otherwise you're kind of rudderless in a new field. And I think that you in fact organize some of these visits?
I used to act as a tout for him and persuade people to go to La Jolla. I said, the food is good, the climate is perfect, and you just have to listen and talk to Francis. But that's how a lot of people got there.
Oh, my goodness, I mean, who would turn down an opportunity like that? But one of them was David Marr.
Correct.
Who came up in your discussion in the last lecture. And David Marr, as you pointed out, had an interesting history. He actually, although he did his thesis on neural modeling, his background was really math.
Yeah.
And he did it with Giles Brindley.
Yeah.
Do you remember that?
Yes, I know. Giles Brindley was a colleague of mine in King's College before he moved to London.
Right. And he was a real professor of physiology, and there was a story I heard, and I've checked it, it's a true story. He had discovered a peptide which was able to support an ejaculation. Not ejaculation, but an erection. Sorry here, I mean, this is a scientific discussion, but here's what he did. At one of the British meetings of urology, he lowered his pants, injected himself, just to prove, as a demonstration, that for the rest of his lecture, it was standing erect. And so he was an unconventional type of person.
He was you wouldn't believe it if you met him. He was quite a small guy, and he was very retiring. But he did a lot of this when he went to London.
I think he even put an electrode into his brain at some point.
He was always experimenting with himself. And David went to him because he'd met him. He was a fellow of King's College. David was a Trinity man, but David got to us because of Horace and the connections that we had, because Hubel used to come and visit us, as did Max Callan in Cambridge. So
David Marr was a very interesting figure historically. He died young of leukemia, but before he died, he had moved to MIT, to the AI lab there, and started a whole new group that was studying models of vision from the bottom up. And so the idea he had, which was very, at the time, pioneering, was that, well, let's follow the photons in. What happens, what goes on in the retina, it gets transformed into spikes that go from the ganglion cells into the cortex, and what's going on. So he had computational ideas and computational models, and he had students working in every little part of the different types of feature detectors and the different types of global edge detection, for example, so segment the object from the background, and wrote a very influential book called Vision. Now, I met David, and this is back when I was a graduate student, at a very small workshop at Jackson Hole, and it's also where I learned how to ski. It was a ski workshop. And David really impressed me because he had charisma. I mean, he really, you were convinced that he's going to do something really important. And so, he invited me to the AI lab where I gave a talk. And it was actually because of him that I met Geoffrey Hinton, who became one of my closest friends and collaborators. And the story here goes way back to 1979 when Jeff he was a postdoc in the Department of Psychology, and he was one of the principal figures for starting what was called parallel distributed processing. David Rumelhart and Jay McClellan were the leaders. He was a postdoc, but he really was the intellectual giant there in terms of thinking about how the brain organizes the parallel processing and thinking about how the interactions between the neurons might compute something very important, recognizing objects and so forth. So, in any case, I was invited to go to this workshop, and who was I? I was just a graduate student, had a couple papers in obscure journals, and it turned out that he had met David Marr and asked David Marr if have you met him, and it was David Marr's recommendation that had Jeff decide, well, let's take a chance on this guy because he looks like he has an interesting background in physics and neuroscience, and it turned out that we really hit it off. We basically believed the same religion. And you really have to you have to have beliefs in the sense of thinking. You know, our view was that, look, let's just take vision, right? It's a very difficult problem. The only existence proof that you can solve the problem is the fact that we can see, right? And the problem is we're so good at it, we don't know what's behind the curtain, all the unconscious parts. And if you just use your intuition, you'll be totally misled, totally. And unfortunately, it turned out that a lot of David Marr's early work was also based on intuition. And although in England, I think they even had a big project, they put a lot of money into it and tried to see how far you can take it. And that really was, it turned out to be a dead end. But nobody could have known that without really going down the road. But Jeff and I really, over the next few years, got very focused, and we started developing what became the Boltzmann machine. Now, the background here is that going back to the early 60s, 59, there was a very influential neural network model called the perceptron. And this was the very first learning algorithm that was demonstrated that it would converge could be used to change the strengths of the inputs to a single neuron so that it could discriminate between different categories. For example, it can discriminate between a tank in an image and of a natural scene and no tank, right? So this was, back in 1960, it was competing with another approach which had been championed by some of the early pioneers of artificial intelligence, Marvin Minsky, who was the partner with Seymour Papert. And AI Lab at MIT was founded by Marvin. And the goal there was to write a program. You write a program where every step is engineered in to be able to discriminate objects. And the idea is you have to figure out what features are unique to different classes of objects. What made this almost impossible was that you have a 3D invariance problem, because as the object moves, the features change, right? Because of the fact that you have different projections. And so two projections of the same object were completely different. Whereas if you have, say, two objects, two faces that are both frontal, they're almost identical. You know? So how do you figure? You know, people would look for fine features and so forth. But make a long story short, there was very, very little progress for many, many years. And there's an anecdote that I came across, which is the very first grant that the AI Lab got was from the military to build a robot that could play ping pong. And they had no idea how difficult that was going to be. But, you know, they figured that, you know, that I can play ping pong, so I should be able to program a computer, because we can program the computer to prove mathematical theorems, right? And so that's the highest form of human cognition, and this is all low-level stuff, you know, that should be very easy. They were misled by the fact that, again, these are things that are evolved over millions of years in animals, right? We are benefiting from all of that evolution. In any case, so they got the grant, and they realized that they forgot to put in money to write a vision program. And so what did they do? The story goes that they assigned it to a graduate student as a summer project. Now, I bumped into Marvin Minsky many years later, and I wanted to confirm this because it's such a wonderful story. So I told him the story, and he shook his head and said, "You got the details all wrong. We did not assign it to a graduate student for a summer program, a summer project. We assigned it to undergraduates." And it is true, the undergraduates generally are smarter than the graduate students at MIT. But, and I've dug into the archives, and I actually have the actual notice saying that they're going to have a summer project for undergraduates to write, you know, they're going to assign each one of the undergraduates as a part of the vision program, right? So, that was a competing approach. It was neural networks versus the symbol processing, rule-based programming. And that was very promising. It was very exciting, and it looked like it was, you know, really, that's where all the money went. That's where all the, you know, the best young people, and the real turning point, because neural networks look promising too, because this is exciting. You can train it by giving it examples. It's a completely different approach. It's actually a lot closer to the way that we learn things, right, from experience. But here's what happened. Marvin Minsky and Seymour Papert wrote a extremely well-written mathematical treatise called Perceptrons. And in it, they analyzed, what is the limit of what the perceptron can learn? And it turned out to be a very relatively restricted class of functions called first-order predicates, and it geometrically corresponds to passing a plane between the examples that you're looking for to categorize and all the rest. And there are some problems that, in fact, that you can solve with that, but the vast majority of the difficult ones couldn't be touched. And at the very end, after it was all this beautiful math, and it was really, really just a wonderful book, and it could have served as the foundation for the whole field. They said at the end that they did not think that this limitation could be overcome with a learning algorithm for a perceptron that had more than one layer, multi-layer perceptrons. And they just said that they didn't think it would be possible. But that stopped research dead.
Yeah.
You couldn't get any money. You couldn't get any jobs. And it really was a whole generation before a couple of naive young graduate students and postdocs like Jeff and me go in and say, hey, wait a second now. Let's give it a try. And that's what we did. And with the Boltzmann machine, which was based on principles from physics, from statistical mechanics, we were able to come up with a learning algorithm that broke the barrier. And demonstrably, we call them hidden units between the sensory inputs and the outputs. There was these hidden units in between, which is the brain, right? That was going to develop representations that would help you solve the problem. And we demonstrated it on some toy problems. The problem back then was that computers were so slow compared to today's. They were a million times slower, so that you could only have very small networks, so we couldn't really scale it up. But the key thing that really got it off the ground was the fact that that we showed that they were wrong, and the floodgates opened, and hundreds of people fled into the field. This is a really exciting time in the 80s. They're coming in from physics, they're coming in from engineering, they're coming in from math, from neuroscience, from cognitive science. And by the way, this had the same feeling that molecular genetics must have had, in the sense that they were all amateurs. Right? I mean, they had their own expertise in established fields, but now they're coming into an area where nobody knew what was going to happen, and so it was very exciting. And we had our national, it was Neural Information Processing Systems meetings. That the first one was back in '87, so it's been going for like 30 years. But it was very exciting, and a tremendous amount of progress was made, and many learning algorithms for multi-layer perceptrons were developed, and the one that has actually become the most popular today is called the backpropagation of error. Now, here's the thing about, I'm coming back now to Jeff Hinton. So Jeff was involved both in Boltzmann machine and in the backprop papers, and he was the great-grandson of George Boole.
Yeah.
And so George Boole is the founder of computer science. He literally wrote Boolean logic, right? That was his discovery. And it really was, that's how computers work. They work with logic. So I went back and read his book, Investigations into the Laws of Thought. And it turns out there's a subtitle to the book in which we apply Boolean logic and probability theory. I said, whoa, probability theory. So I took a look, and sure enough, several chapters in the book toward the end weren't about logic, which is, black and white, but it was about probabilities. Well, it turns out that the probabilities are really the foundation of machine learning, from which neural networks now are one of the most powerful algorithms. And it's all about probability distributions, because the real world is not black and white, except your scarf. It is combining information and coming up with a good guess, and it has to be done very quickly. You don't want to do it, spend all of your time computing away and at the very end come up with the wrong answer. You want a reasonably good effort because good guess because your life might depend on making a decision, right? And you have a tiger there. What are you going to do? Should you run? Should you just freeze? You have to make a decision. And so, in some ways, this is really remarkable. George Boole was, in a sense, the father of both traditional logic-based AI and machine learning. And I'm sure that he would have been very proud of Jeff, right, his progeny, that was involved in that. So this is, and this actually goes back and this reminds me of something else that makes contact. Do you know who his thesis advisor was? Jeff Hinton?
No.
Christopher Longwood Higgins.
Of course. You see, and I was about to add another person that was very important in David Marr. It's a man called Swinnerton Dyer. This man, he rose to be the head of the university and a big shot in education. But he was a mathematician, a very good one. He realized hanging around the lab that you have to realize when these computers, the first two computers, called ETSAC 1 and ETSAC 2, were built in Cambridge. ETSAC 2, I remember, it was a 17,000 valve machine.
That's a vacuum tube.
Yes. And it actually calculated the first Fourier of myoglobin.
Wow.
But all of us, we used to go there because when it worked in the summer, it got very hot. It wasn't cooled at all. And we were each given a comic book to stand and wave. And the machine would work and then a valve would go, had to be replaced. So we used to go there in the middle of the night when these calculations were being done. At Swinnerton Dyer, he noticed it, and he decided one weekend to write a programming language for this machine. So it was the first machine for which there was a programming. It predated, it was just after the war. There was only one other big computer. That's the one that von Neumann built for the military. But it's, you see, there were all these connections. I mean, Turing himself was a don in the same college that I was in. Turing was a don there. He went to Princeton, and then, of course, he would then work during the war. He's the one who broke the German code. But they were very careful not to let on that they could break these codes. In fact, they deliberately left certain things happen, because if they'd have known they knew that there was so there was a little bit of an avoidance, but they didn't want them to know that this great code had been broken. Swinnerton Dyer worked with that group, as did another man called Donald Mickey, who went into AI. So this is after the war. There was a whole complex of people had nothing to do with their formal education in any way, but they actually invented the new fields. And I think that for those times when a field is just starting, that I think this is very productive. And the worst thing that happens when it gets settled down into very programmatic and then gets a structure, you know, like people have 65, 70 people in a group. Now, of course, some of the big installations have more than 1,000 in the group. If you look at papers today in physics, you will find that the list of authors is several times longer than the paper itself, which is just some incomprehensible thing full of Greek letters that's supposed to be important. However, so one of the things that one should always try to do, and which I think is something we achieved in Cambridge, at many labs that descended from ours, was always the same thing, which is just everybody should have, which I said the other day, their own project for which they are entirely responsible. And if they don't do that, then, you know, that is the test. And if you are successful there, you know, exactly what you've studied doesn't really matter. You've learned how to solve problems. Well, I think that the major problems still exist in biology. I remember we said, oh, well, we're just going to leave molecular biology because now there'll be hordes of people, they'll solve all the crystals, they'll solve all the things, they'll just turn the crank. So that's why we got out of it. And people said, how could you leave it? It was just, yeah, but I mean, what's the point? What's the point? In fact, if it hadn't been for Francis Crick, I would never have written papers, because I thought once I've solved the problem, can't I put a notice somewhere? Why have to write all this and go through with referees and so on? But he used to lock me up in the room, never let me out till I completed the paper. But I think that there was nothing that worked like this here. The two of us, we rarely wrote a paper together, one or two. But everything was discussed as to what you should do, how you should do it. And we then involve everybody else in this. But nobody was allowed to, you know, start to make a hierarchical group that X would do this and Y would do that. So I think that I was, of course, much more experimentalist. I like working with my hands. I think it's great. And Francis, of course, was a theoretician. But I still have many of drafts of that Francis did. They're all in Cold Spring Harbor, because another bad habit of mine was never to throw any paper out. Just keep everything. You never know when you might need it.
So it's interesting because this is about a year ago. I was tracking down some paper, and I actually hit. They have it online. Your letters.
Yeah.
And it turned out that a review that you wrote for some Nature. Yeah. Was in this archive, and it was really funny to see this, and you know that you were you know, going back and forth with the editor about whether or not the paper it should stand. By the way, with regard to your decision to go into C. elegans, so I've always, and you said this must have been a big. You started out by hiring a few people, but
no, I started out by reading. I read about all things. I had to find out about this, and I told them, see, what was very important was a great zoologist called Richard Goldschmidt. As a student in Munich, he became a professor of genetics in California, if I remember. But he, as an 18-year-old student, did the nervous system of Ascaris, which is a big worm. And when I went to read the paper, this was in a German, it's in 1911 is when he published it. The pages had not been cut, which means that it sat in the zoology library in Cambridge since the early part of the century. No one had ever opened that journal. But that was good. Plus a book which I've had sent. It'll be in the library, which is a marvelous book of people about the nervous system of different animals. That's really great. And you can see what people were looking at, all kinds of preparations, they were called.
So you must have quite a collection, because you told me a story. This just occurred to me. You told me the story that you wanted to buy J.Z. Young's book about the octopus.
Oh yes.
And so it was out of print, so you sent off to.
I've got that book. That book is coming here as well.
Okay.
So,
but the story is, though, that when you got it, you picked it up and there was a frontispiece, property of Salk Institute.
Yeah. See, what had happened is I wanted to get J.Z. Young, was a pretty good zoologist. And I got interested in this invertebrate, which has as big a nervous system as a mouse. It's got 10 to the 9 neurons and is very smart, as people have discovered, and is the kind of local genius of the invertebrates. Of course, it has to live underwater, so it couldn't do many things. But I remembered that professor that I knew, J.Z. Young, I had all of his books, but I didn't have that book. It's called The Nervous System of the Octopus. And so I looked it up in Amazon, and there was a copy, $75. So I wrote away, I got it. When it came, I opened it, and in the frontispiece it said, property of the Salk Institute Library. The library had begun to throw out books. I used to go regularly to the outside and get books. You could help yourself. Classics. But of course, you see, that's what happens when things are old. They just get thrown out. I just want to tell you a little story about a very pretentious Frenchman called Jacques Monod. Jacques Monod was a very sophisticated French scientist. Is well known for induced enzyme and for writing many of the classic things on allostery. We used to say that's how you answer the phone. In Paris, you say, "Allô, Styrie?" So anyway, so Jacques Monod, I once came to the Pasteur Institute, and he says, I've got to tell you something. I go to his room, he's written on the board. He says, repressor, not repressor, equals inducer. He says, that is the logic. So I thought I'd just been rather naughty. I said, you know, Jacques, I think I've read something like that. It's done by an English scientist. I can't remember his name. I think it was Boyle. No, it might have been Boole. It's the laws
Logic.
of thought. I went down to the bookshop down there in Paris, and I bought, there was a Dover edition of it, and I left it on his desk to say, there you are. In other words, that sort of background of education that we got automatically, you know, by studying all of these things, they had never heard of this. Philosophers were something different in France. Anyway, I think that I've got another copy of the Boole here, and Boolean logic is very important part of this whole development.
So to go back, this one question I just wanted to ask, who gave you the first grant for the C. elegans project? Where did the money come from?
I'll tell you what happened. We had formed, you see, there was a group in the Cavendish laboratory. That was the original group, with Francis, myself, and the protein crystallographers. We were all together in the Cavendish. In fact, we had got to such a stage that we'd been doing genetics there, that Francis was our great seeker of space. So when we became very adept at finding storerooms and then asking the professor, can we use that room and convert them on a weekend to a lab? So that there was a huge long room, about as long as this. It was in the grounds of the Cavendish lab near zoology. It used to house a whale, the skeleton of a whale. That finally decided to get rid of this, so there was this long room, just wide enough to have one bench by the window. And so Francis got us to use that. So we borrowed it from old zoology, took us a weekend, and we put all of our people. And it used to be very nice because you would sit, the window was in front of you, and you had a courtyard. And quite often, you know, you'd see an elderly gentleman walk through there, and you'd take these Americans and say, that is Dirac. You know, all these really giants, they used to walk through this thing to go through the courtyard. And we did a lot of work there. Eventually, it just became impossible to house what we wanted to do. Fred Sanger, who had a Nobel Prize already for insulin sequence, was working in the biochemistry department. I started working very early with Fred. And Fred is another unique person. If you want a little bit of a flavor, if you read my obituary of him in Nature when he died, I think it was last year, just have a look at that. Because Fred was absolutely untypical scientist. He always worked with one technician in a tiny room himself, always with his own hands. never employed very many people. And he developed the whole of nucleic acid sequencing. So we said we want to go together with Fred and start this thing. And it was the first time we decided to call it a laboratory. That's why it's the only thing in the Medical Research Council that isn't a unit or an institute. It's called the Laboratory of Molecular Biology. And you'll notice that's what I call everything I try to work in is called a laboratory, because these institutes are not the things that we had. We had no managers. And so the MRC thought they then produced the money. But they first wanted a MRC institute to house a whole lot of MRC. But Francis said, we don't want them. They're no good. We just want to go with this. And we then recruited some people from London, came to join us. But I think that was the thing. The money ran out before my lab was actually had this, a lot of the things were wired up ourselves, because when the money ran out, it ran out. Anyway, that's how we started to go there, and we always kept the same style of organization, always. And it is also a very healthy way of doing science. You keep contacts with everybody. Everybody is collaborative. And nobody's scared of doing new subjects. If you need to start, you just start. And you can put it together in a way and get going. And so that's the way we did everything there. And that's a kind of organization that Terry will amaze me, because will amaze you, because we managed to get Francis was very influential in getting the Salk Institute, getting the man who was the head of a big company.
Irwin Jacobs.
Irwin Jacobs. He later became the chairman of the board. But we said we want to just have a special little group, which we formulated. But the Salk Institute is the president owns the money, but the actual academic group own the appointments. And when they can't agree, nothing happens. So we were waiting for this group to get going. And I remember coming in and said, right, Terry, you're appointed. Now, as of today, you're the director of this. By the way, it was called after Francis Crick and Irwin Jacobs, but it's called the Crick-Jacobs Laboratory, and not the Jacobs-Crick, which is a cheap Australian wine, if some of you know. I made very clear it had to be called Crick-Jacobs. Otherwise, we'd be confused with cheap Australian wine. And Terry became, and we proceeded to hire young people and let them go. And we found some really great talents, many of whom have stayed on, they've gone elsewhere. But that was a really productive period.
So one of our junior fellows was Gene Yo,
There's Genio,
who was well
came
known
there.
here. He's now a professor at UCSD.
Yeah.
Philip
And you-
Yo's son, in case you don't remember.
You see, so Gene I got out of MIT by giving him the advice, you see. I said the aim as a graduate student is to be moderately successful. Why? Because if you're a failure, your supervisor will kick you out. If you're very good, he will keep you there for 7 or 8 years. But I got Gene to go and get out after 4 years, which is a record, to just get away so he could. And then he came to the Salk, and, and I think just having that opportunity to get into science, to create your own thing, is very good.
This might be a good place to take questions. This might be a good place where we could take questions.
Yeah.
While people are thinking a question, just want to. You asked what an English was to be fired.
Yes. What's the word for that?
Made redundant.
What is this? I didn't hear.
It was
What was it? Repeat it.
earlier. You were saying you couldn't remember what in England they would tell you if you were fired. You would be made redundant.
Oh, made redundant.
Oh, yes.
Yes. Okay, that's the euphemism. Yep.
Yes. That's a euphemism.
Okay. So you mentioned that you got the funding for the C. elegans project in England. However, the research on C. elegans had sort of like started a little before you started it, right? Like you mentioned Dr. Ellsworth
No, there were
Dougherty.
people. There was a man called Dougherty
Yeah.
at who started this, and a Frenchman called Nigon who started it in 1929, working with this. They were curiosities. Dougherty's great aim was to grow them in defined medium. He never succeeded. And it just became very thin. But they already knew enough to see you could do really good genetics. And we knew enough from what had been studied to know that it had a small number of cells, many differentiated tissues. In fact, as we used to say, it's a real animal. But it was a great subject of plant pathology. Nematodes eat almost everything. And of course, they're also infestations of humans. They're parasites. So I think it wasn't an empty field by any means. And that's when I went on sabbatical to California in 1964. It was to get, no, 62. It was to make the break, because it's very hard to stop doing things in one lab. I really wanted to go and do something completely different. So I think I still stayed back 1 month to start some new molecular biology, which was on transfer RNA, where we learned how to sequence it and did a lot of things. And nearly all the students who came to work with me started on transfer RNA, and many of them came from molecular biology to come and work on C. elegans. And that's how we got streams of very talented young people into the field.
Where did your first worm come from?
I got it. I collected 52 worms from nature. 2 of them, called N2 and N52, they came from Nigon and Doherty. That's the C. elegans. But there's another one which was quite common called C. briggsii. Many have now reintroduced those, but are isolated. I had a friend who was in charge of an oil company research thing. And so I got soil sent from all countries, 5 degrees from the equator. I thought I'd get something that would grow faster at a higher temperature. Never succeeded. But I had a lot of them, and I had a vast collection, but they were all now gone. We did a lot of exploration on how to fix them, cut sections of them, but all of that was really preparation. But after the methods had been developed, became very easy to start. My first mutant is isolated in 1967. All the notebooks are in Cold Spring Harbor. You can just go through, because we wanted there to be a communal notebook. So everything you did, you wrote up, you put in these books, the loose leaf binders, and they went up to mutant 1200 or something. So it's a big collection, but you could track back what everybody had done, what observations they'd made. So, but of course, after a while, just got too much.
Yeah. So you mentioned N2 and N52.
Yeah.
What happened to N52? Because
That is the Nigon thing. I think it got kept somewhere.
Okay.
There is someone who isolated them. There's even one got isolated in Romania. They're very common, these Caenorhabditis.
right, I'm asking because nowadays people use N2 as a wild type strain in C. elegans, and that's the only one exists.
Well, there's an N52, I think is still probably, you see, we discovered how to preserve them.
Yeah.
They had to be frozen and kept in liquid nitrogen. So there were all these liquid nitrogen tanks with all the reservoirs of mutants, and you could just revive them, you see.
As long as you don't drop them first.
So you could do that. So this meant that the collection was always, if a new student came, like Jonathan Hodgkin, we could just give him something to do, and he would start off his own thing. And everybody was allowed to take back their study, to go and continue with it. It's just a vast to try and do total biology. It just says, it's now, I think at the height, I think there were 4,000 scientists working, mostly in America, the UK, but it spread to Germany, Japan. So C. elegans just became a kind of standard object now.
Thank you.
Hi, Sydney, you mentioned that there are some major questions in biology that are unanswered. I'm just wondering what might those be? And what might be
Major questions that are unanswered in biology.
which you
That
mentioned?
are unanswered.
Right.
I think they're all to do with the nervous system.
Okay, and you also mentioned that there were times in your life where you thought you saw the light. And I'm just wondering, what are some guiding principles that you thought could help bring us closer to sometimes experiencing what you have experienced?
Well, many of the things where you can suddenly see through, there's a famous thing's been written up, was just remembering one tiny little fact can sometimes say, you know, it used to be, as we used to say, Francis Crick. So, but I think that that's a unique time. Most of the thing is quite a business to get it together. And I can't write about anything unless I've got it completely clear in my own head. If there's some problem you can see, I try to write around it, but I just can't get past that point. So I think you just have to have the whole thing clear in your head and not something that you can do, you see. And I think, evolution is one of those subjects for which, there's been a lot of discussion. There's a lot of information available on this. But to try and put the whole of life together, the last 2 billion years, into one stream, that's always been an intention of mine. But of course you can't do it, because of all the steps of going from single cells to multiple cells, all the different kinds of animals. We used to say there are 3 kinds of higher organisms. There are vertebrates, there are invertebrates, and then there are pervertebrates, which are things that are neither there. But I think it is all coming together. It is all coming together. And I think we can begin to see, for all the arguments that have been, and they are still with us, the importance of selection versus, people are trying to find clocks that measure the passage of time, but we've now solved that one. It's very interesting. There are things, because of the way time is counted in evolution, is counted as mutation rates. So you will find when you, perhaps I'll give a talk later on this year, that when you come, that mutation rates are measured in generations. All right? So plants already have reached the equilibrium. That is, plants have reached the stationary phase of the Markov process of single base substitutions. So trees, which don't reproduce so rapidly, because the plant grows and reproduces each year. Okay, so, and they've already reached this. Certain animals have reached the stage, either because they're very old and live fast, if you like, but certain animals are still quite young. and are still developing, you see, the distribution of mutations that you can measure, you can quantitate it. So we can predict, see, the mouse's life period is only about 3 years, 2 to 3 years. So mice, you have to believe me when I tell you that they're over 100 million years into the future already. The mouse genome's pretty good. It's already because it's evolving 2.5 times faster than a slow genome like ours, or even slower ones that exist. See, there are animals that die and reproduce only 100 years, roughly speaking. The whale shark has just been done. That looks like a very young genome, hasn't had much time, even though it started 460 million years ago. Still is quite an immature genome. Still got a long way to go. So you have to have, so the idea now is you can take, and I just mentioned that you can take a part of the genome and you can so define it that every substitution, mutation there, leaves all the proteins unaltered. Okay? So it's a real clock. All right? And you can measure how fast this clock is ticking in all the animals, plants, bacteria, and you can tell how fast it's clicking, and you can then make a prediction of when it will come to reach the equilibrium. All right? And that is something that, with this big discussion between Darwin and all the other people who were against that, you see, it was nonsense. It's both. But don't get confused. As it is that most of the changes that are recorded have no effect on the organism. They're just the clock ticking. So once you can isolate this, you can look at different organisms just as a set of clocks. And you can count them very easily as to how far they've got, because they convert steadily the Poisson distribution. It's very strict. It is a Poisson. You can prove that. And they convert that into the so-called normal distribution. And once that reaches a steady state, then as many genes are going backwards as are going forwards. So you just stay in the same position. It's just balanced by forward and reverse mutation rate. So that's a simple invariant if you do this. And that gives you some very interesting things. So it also gets a little confusing to talk about younger and older genomes, because, of course, when we talk about evolution, we're not interested in this regular clock. That goes on, and it's just a property of the organism. And in fact, you can show that it's shared by yeast, Drosophila, anything shares this clock. What we want to know is, how did octopuses change into mice? That's the real question, you see. How did people become what they are today? I mean, it's quite a short time in these terms.
you know,
See.
Okay.
Is it on? Okay, this question is for Terry. Do I have to stand up? Okay, hi. So I was wondering, is there anything about the current so-called AI revolution that surprises you? And how far do you think we are from like a digital avatar that can behave like ourselves?
Okay, well, that's a very good question that will require another discussion and tomorrow. But I'll tell you, I have written a book with exactly that title, The Deep Learning Revolution, so I have a lot to say.
We
All
should
right.
talk about that.
This was a very short question. Then there's room for one more little. Yes, Brian?
Yeah, I was struck both your cases that you kind of followed your intuitions, you followed your own initiative, your own imaginations. And that's, no, I'm an outsider here and I say this a little bit carefully, but that's not the tradition so much in Singapore. I was brought out by some civil servants a year or two ago, and I asked them why they wanted to talk to me, and they said, we heard you got the first research going at the Santa Fe Institute. I was in charge at that time of research. How did you choose the projects? That's not what I'm hearing here. So I'm wondering what you have to say about being a self-starter versus part of a hierarchy where research projects might be given to you.
Well, okay, so I'll take the first crack at that. So you're absolutely right that we were rebels, but with a cause. And you know, Jeff and I basically really realized that we had the same intuition. By the way, he came to the U.S. because the Lighthill report.
Yeah, that, you see, that killed.
That's top down. They killed AI.
They killed AI
in
England.
Yeah. And so, you know, there was, still at a low level, but it was still possible to do the research in the U.S. And I think that it's just the same story you heard from Sydney, a small group of us, literally maybe a dozen. And we would get together in these little workshops, and we had so much fun. I mean, it was just so great. And you know, we didn't really know for sure where it would head, but we, because of the fact that we were naive, you know, we didn't listen to the experts who told us that it was impossible. We just went ahead and did it, and that's really what it takes. And it's something that you know, when you're young, you can take these risks. Or at least, you know, you're foolish enough to take these risks. Oh, I have to. It's just one story. Okay. My first grant. Jeff Hinton was in England at the time. He was doing a postdoc at the MRC in Cambridge. Not MRC. It was it was one of these British institutes in Cambridge. And it was not the. It was and he gets a phone call 3 a.m. from Charles Smith, who identifies himself as the president of the System Development Foundation.
Yeah.
And he explains to Jeff that their foundation is trying to identify people who are doing risky research that's unlikely to succeed. And they had gotten Jeff's name. Now, he wasn't sure whether this was someone was joking, but you know, it's like, you know, was it the Pascal's bet? You know, well, yeah. I'm going to go along with it, and so Jeff said, well, you know, I have a friend, Terry Sejnowski, whose research is even less likely to succeed than mine. And lo and behold, we both got these big grants to start our careers. Mine at Johns Hopkins and his at Carnegie Mellon. So, you know, it depends on these things that fall from the sky that you can't predict.
Whether it works or not. I think we should thank both Terry and Sydney once again for a wonderful lecture. Tomorrow we'll have the last of the lectures. That is going to be your last chance to ask all the questions that you have collected over the last 3 sessions. There will also be a discussion like today. I don't know on what, but I'm sure that you know.
We'll figure it out.
You'll figure it out. It's going to be very interesting.
We're not going to prepare anything.
Very good, very good. So I invite you to have a cup of coffee and something nice in the back. Terry, he will be around. I think Sydney will probably
I have to go and get my feet up.
All right.
Or else the doctors will give me up.
Yeah, that's not a good idea.
Okay.
So so please enjoy yourself. Attack Terry on anything you want to. Have something to eat, and we'll see you tomorrow at 12:30, 1 o'clock.
Yeah.
Thank you.